Okay, everyone, hang in there. We're going for a ride. Neurophysiology, what I consider to be probably the most challenging part of material for this course, for those of you who haven't had a lot of courses with cell biology and so forth. We need to know some of this so that we can understand why things are happening in the brain and why some of the symptoms of mental disorders are actually being caused. So you will see that this is important later on down the road. And so here we go. We are going to start sharing these notes. Now, first off, I do want to say that this is not a PowerPoint presentation. I added a lot of text and I just wanted to revise things and get as much as I can in here. So please forgive me for not having that for you. I want to show you this second hyperlink here, and don't laugh. It says Neuroscience for Kids. This is one of the most awesome sites, and I don't care how old you are. The first link takes you to the basic index for this Neuroscience for Kids. The second link takes you to this about the action potential. And I just love how they set it up here. You're going to be able to see exactly what's happening at different points. And I think this is so important. And this little interesting fact, the giant axon of the squid, which is where we learned all about this, can be 100 to 1,000 times larger than a mammalian axon. Now think about it. That's a lot bigger. That's one axon. And so that is really how we learned a lot about it. Now, there's a lot of animations that are here that normally if I'm in class, I will bring a couple of them up and go through with you. But because it's so important to me right now to get through this material, I'm going to leave this for you to do for now. Later on, maybe we have a chance to come back. I love interactive stuff, so one of the reasons I really like this. So let's go back and remember, think about what the membrane of a neuron's cell, that cell membrane is. That phospholipid bilayer. And remember that charged molecules can't just go whipping through, all right? They can, however, go through other places. So what we need to do, those other places, you'll see what I mean in a minute. Let's get set up for the resting membrane potential. And right at the beginning, I want to tell you that the resting membrane potential actually requires energy. So first, we've got to have cytosol, which is intracellular fluid, the fluid that's within the cell. And we also need extracellular fluid, which is their fluid on the outside of the cell. And what are the ingredients within these fluids? Water, obviously. Remember what water is, H2O? It's one oxygen with two negative charges and two hydrogens coming off at angles, which are positive charges. And so we have an uneven distribution of electrical charge. So anything that is charged, that is in the vicinity, remember charge means polar, is going to be attracted to either the head or the fingers, the positive or the negative side. And bats are not charged, so they don't like the water. They're hydrophobic, remember? But the bottom line is with this, molecules are going to split up and go to those specific ends of the water. One reason we use it for cleaning. Ions are also essential. These are within the cytosol and extracellular fluid. And What is an ion? It is an atom or a molecule that has a net electrical charge, meaning that if you add up the electrical charges, it's going to be not even. It's going to be either positive in the end or negative. And if the ion has more positive, is a positive ion, then it is a cation. Sodium, potassium, calcium. Those are important ions that we'll be talking about in reference to that resting membrane potential. Now, what if the ion is negative? Think of negative, take away from, that's an anion. An a in front of a word, a prefix, means take away from or without. So chloride is the ion that we're going to talk about that is right now an anion. So what else do we need besides the cytosol with these ingredients? We need that phospholipid membrane. See if I can do this without flipping us down too far. The phospholipid membrane, remember we have a phosphate group portion that is polar. That's this head. And remember, that is like hydrophilic. I love water. Let me be next to you, water. Whether it's outside of the cell or inside the cell. Doesn't matter. It likes the water. So the hydrocarbon is nonpolar. That's the tail. This phosphate group that I mentioned already is hydrophilic and loves the water. So the tails, or hydrocarbon, nonpolar part, is going, keep me away from the water, whether it's outside or inside the cell. Don't want to be there. That results in a stable arrangement of two layers, that bilayer. And this serves to form a barrier to water-soluble ions. Remember, this is water-soluble ions. One of the reasons that a lot of things can't get into cells. Those are charged molecules. Question, you should be thinking, well, something's got through, so how does that happen? We'll talk about that later. So we have also the need for proteins. Proteins are essential for forming the enzymes we're going to need, for forming the cytoskeleton, which helps maintain a shape of the cell, and receptors. This is one area we're going to be spending some time on, receptors. And the proteins, if you recall back from a previous lecture, are synthesized in the ribosomes, the rough ER, endoplasmic reticulum, those R's, ribosomes. And those are built from amino acids. In the membrane, we have these proteins embedded, and there's two very different types of proteins that turn into receptors. They're channel proteins and signaling proteins. So as we go on down, hopefully you've got that, we're going to start discussing the resting membrane potential. Now first, what's potential? I'm pretty sure you've all heard this at some point in your life. Oh, you have a lot of potential, or you're not living up to your potential. So what is potential? Potential refers to a difference, right, between one place and another place. In your case, it's like somebody might have told you that this is where you are, but you can get over here, and that's different. In this case, we're talking about the difference in electrical charge between the inside of the cell, cytosol, and the outside of the cell. And we can actually measure this difference with microelectrodes, little itty bitty tiny electrodes that can go into, one will go into the cytosol and one will be in the extracellular fluid. And these microelectrodes will send information to an oscilloscope, and that will give us the difference between the two. How is the charge in here different from the charge out here. Now, when we put this neuron in a resting state, there is a difference. It is polarized. The difference amounts to minus 70 millivolts or minus 65 in there. And that means that the inside is more negative than the outside. This will make more sense in a couple of minutes. This is important, and it's setting us up for the action potential eventually. And without an action potential, just looking ahead, there is no activity of the neuron, there's no neurotransmission. We can't release neurotransmitters. So we have to have these action potentials. So there are 4 factors that are involved in the maintenance of a resting membrane potential. And I'm making a big deal of this because guess what? You're going to need to know these 4 factors for maybe quizzes, maybe oral exams, maybe written exams, but definitely we need to know these. So to have a resting membrane potential, or to maintain this difference in the millivolts inside versus outside of the cell, we need these 4 things. First, selective permeability, which simply means, right, some things flow through, others don't. And the main players in this picture of selective permeability are ions. Remember those ions. And because of the setup, we have more sodium on the outside. This membrane permits only a little bit of the sodium to leak in. And there's more potassium on the inside. And the membrane permits only a little to leak out. The chloride that we see here is mostly found outside. Now, large protein molecules inside are anions, and they're part of the cell's machinery. They won't go through the membrane, okay? So the end result is, let's look up here again, is a net negative. Look at how negative these guys are. Whoa. And we'll review this in a bit, in a different way of thinking about it, just to kind of give you a couple ways to remember this. So that was factor 1, selective permeability. Factor 2, sodium potassium pump. These are literal pumps that, guess what, have the ability to move sodium and potassium across that cell membrane. They are pumps, therefore they require energy. And in fact, these sodium potassium pumps actually need about a half of the total cell's energy. And what they do is they take 3 sodiums that have actually leaked inside. Okay, they've leaked through inside the cell. That's down here where you can't see. And brings and throws them out. But it grabs 2 potassium that have leaked outside and brings those back in. So think about, you've got 3 positives going out, 2 positives coming in. So that, remember, this positive makes a huge difference. This generates a potential. Since you're sending out 3 positive charges and bringing in 2 positive charges. So you support a net negative charge inside the cell. Remember, you're sending positive things out at a higher rate than bringing them in. So that's going to help maintain a more negative environment within the cell. But think about it. You've grabbed these potassium ions, and now they're all sitting around inside the cell. Oh man, this is a little boring. Wow, wow, wow. And that leads us to our third factor in maintaining the resting membrane potential, and that is the concentration gradient. You guys all know about this. Brownian movement basically states that any molecule that is warmer than absolute zero is vibrating randomly, okay? And if you have 1,000 potassiums, I'm just going to say potassiums is shorter than ions and everything, inside, and only 100 outside, where are they likely to move? This particular ion. You're like, wow, we're really crowded in here. We're going to move out. And it is because we have this natural concentration gradient that we even need that sodium potassium pump. Okay, so now more potassium will have a tendency to leak out, and that further promotes a net negative charge inside the cell. We're getting rid of some of the positivity, if you will. So that's three. We have selective permeability, we have the sodium potassium pump, and the concentration gradient. Being that we're talking a lot about electrical, like voltage, positive, negative charges, it should be pretty natural to move on to the idea that the fourth, whoops, sorry, I didn't move these over. The electrical gradient is the fourth factor. And because we have a negative charge inside the cell, oh, we're negative in here, it will attract some of the potassium back in because of that electrical gradient. So there's really, and the bottom line is a compromise between the concentration and the electrical gradients. And this negative 70 millivolts really, in essence, reflects the ability of potassium ions to seek out equilibrium. It's like, oh, where should I go? Where should I go? So we're trying to get this equilibrium. And again, as I mentioned before, this requires a lot of energy. Why are we going to expend energy to maintain a resting membrane potential? Well, first, we've got a setup that now will allow for a very fast response, like the trigger of a gun. Like if you pull it back a little, it might not engage. I don't know, I'm not like a gun person, but little I know. But once you get to a certain point and you pull it, it's fast. It's not like a bullet would go, okay, I'll get there someday. So we have a fast response, boom. And it allows for the amplification of signals. That's important to think about. You have all these ions in there, just flip a switch and boom, thousands flow out. Okay, so it is a amplification. If you had a few people inside a building and then you started cramming more in and everybody's going, and all of a sudden they come out and they're all allowed to shout as much as they want, as loud as they want, it would be amplified from what it was before. So those are the two basic reasons, and you can imagine you will need to know those for some of the assessments coming up. Equilibrium is established such that there is no net movement of ions across the membrane, leaving a charge difference between the two sides. Remember, the net movement means there's no movement either way that's great enough to alter the charge difference between the two sides. So here we go. We're going on to generation of the action potential. It is what we call all or none. It always results in the same distribution of ions outside of the cell. Remember, I'm kind of using down here as inside the cell, regardless of the size of the stimulus. And go back to that trigger of the gun. If you squeeze harder, it's not going to change how the bullet goes. Squeezing harder, squeezing weaker, once it hits that one point, it is going to result in the same outcome. And remember that about protein channels, we have ion channels. Those are the channels where sodium can come through. Even though it's charged, there are these protein channels embedded in that cell membrane. Remember, I talked about that earlier, okay? And so these are ion channels. Now, hang on with me here for a minute because it's easier to go through this instead of going back and forth, at least I think, between diagrams and then come back to this and then back to diagrams and back to this. So I hope this works for you. You can always go back to this section of the video, hopefully, and review if necessary. Okay, remember how when we talked about a neuron's anatomy, the external anatomy, there was that cell body. Let me put my hands back here. They're going to look giant. Cell body, and at the junction of the cell body and the axon, we have that axon hillock. And that place, that particular junction, basically summates all the excitatory and inhibitory signals received by the cell. And again, I will talk about this with a diagram in a few minutes. It is here at the site of the axon hillock, important, that the protein channels that are sodium channels embedded in the cell membrane undergo configurational changes. It's like, oh, when it's excitatory enough, it's like, whoa, I'm gonna do this, I'm gonna change, I'm gonna open up. And when there is enough positive charge, these open, and that's going to result in some influx of, you guessed it, sodium. These are called voltage-gated channels. Now, we now have changes in the polarity. That is depolarization. And this is a word that sometimes people take a while to get used to what it means. Remember that polarized means different. You're different. Different from where you are and where you can be, different, and negativity to positivity, there's a difference. So if you depolarize, you're taking away that difference, you're minimizing the difference, you're bringing it closer together. So when more positive charge comes in, remember how that membrane potential was -70, maybe -65. It is reduced, that looks like it comes out, to only -55. There's only 55 millivolts difference between the outside and the inside instead of 65 or 70. And please, don't get nervous if you're like, oh man, I can't remember this. I've got some ways I'm going to go through this again to hopefully bring it in with a different imagery, let's say. So the membrane potential, as I said, goes to -55. So it's reduced, it's depolarized, it's not as different. And that is what we consider the trigger threshold. I need to make a change. Okay, sodium flows in. So there is the influx of sodium ions into the cell because these sodium channels or ion channels have undergone configurational changes, opened up and go, leave room leaves room for that sodium to come on through, go into the cell and have a party, okay? Now, the voltage-gated potassium channels, oh wow, we have other protein channels, you better believe it. And these are voltage-gated too, and they're specifically for potassium. It's like, potassium, I'm a potassium channel, come out here if you're potassium. When they open, after this happens, potassium that's near that membrane are driven out. So we have an efflux, a moving out of potassium ions. Now, after this happens in just 1 millisecond, that's not long, the sodium channels will close. The doors close. Those channels close. However, there's more potassium leaving. Okay, let's think about it. No more sodium bringing positive charge in. Potassium with a positive charge flowing out. Oh my gosh. We are getting closer to being more negative on the inside. That is called repolarization, which is making different again. Now we're going to be more negative on the inside, more positive on the outside, because no longer do we have sodium flowing through. It's staying out, maintaining positive on the outside. And we have potassium still going out, taking positivity out there. So we're getting back to the polarization that we had before, called repolarizing. Okay, it's repolarization. Now, it takes a while for these particular potassium gates to close and for everything to return to normal. And for a while, we become so negative on the inside. We've lost potassium ions. They were so positive. They're outside. We have no more positive sodiums coming in. So now the inside gets more negative, and it goes all the way down to minus 90 millivolts. So now, instead of only 70 or 65 millivolts difference, we have 90. Remember that it's primarily potassium efflux leaving the neuron that returns that axon hillock to its resting potential. Okay, so hang in there. Here is a little illustration, a diagram we're going to go through. Here's the neuron, and there's going to be more of these illustrations coming up. Sodium actually enters into the inside of the axon, right, through this ion gated channel. The sodium protein channel opens right up. And this charge basically spreads. It goes kind of downstream. It goes bump, ba-dum, 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 gets to this sodium channel, and it excites it, and this sodium channel changes its configuration, opens up, which will allow more sodium to come in. Again, why? Because there's more sodium on the outside. And this will go da-dum, 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 go on down the axon. Pretty sure it doesn't say dum-da-dum, but anyway. And so these voltage-gated channels open in response to that depolarization, okay? Now, oops. I want to talk a little bit about the refractory periods. I'm just showing you ahead a little bit. And up through the conduction. And then I'm going to come back with some different ways of thinking about these, okay? I promise it's going to get easier once you put together what we've just gone through with some illustrations I'm going to come up with. Okay. So the refractory period. We have 2 refractory periods. What that means is time when there can't be another action potential. There can't be this excitement. There can't be the sodium gates opening. Okay? And that is for about 1 to 2 milliseconds after that action potential. There just can't be another one. I kind of liken it to my -80 freezer in my lab, that it wants to maintain this really cold temperature. So if you open the door and take things out and then shut the door, there is a couple of seconds or a few seconds where no matter what you do, that door does not want to open. It's like super sealed to try to bring the freezer back down to its temperature that it's supposed to be at. Okay. So during this absolute refractory period, sodium gates will not open. And the fastest that a neuron can fire then, okay, because of this, is 1,000 times a second. Actually, most fire only between like 10 to 200 times per second. Now, there's a period of time when my freezer will open. I just have to pull harder on it, but it will open. That is a relative refractory period. In this case, it needs more than the usual stimulus to initiate another action potential because of hyperpolarization. But it is possible. You just need to send a lot more of excitatory information to that cell and have it picked up by the cell body, dendrites, and so forth. It is possible. You just have to work harder. Okay, before we go on to the next section, I just want to step back for a minute and go through again those 4 factors that are involved in the resting membrane potential. And I want to give you an illustration that is something that maybe, you can visualize more readily. Let us say that there is an establishment in Buffalo, let's just say Kohl's, someplace like that, okay? Whatever you want, whatever will stick in your mind. Okay, that is our cell. Now, we have doors, right? And those doors, to get through, you should be legal. Okay, you should have official identification that tells you and them that you're 21 or older. And so those who have legal documentation in terms of age are our potassium ions. They can go in, they can hang out in there. They can hang out as long as they want. They're in there, yay, having a nice time. We're pretty positive in here, but we've got some negative things too, which we'll talk about in a minute. Okay, but you said, what about the sodiums? Well, there's more on the outside of the bar, right? Why? Because they aren't 21 yet. They aren't allowed in because they can't just move in with their age, their license, whatever. So more of them are on the outside. And some of them sneak through. Can you believe it? Some of them have fake ID. And they'll get through the door and they'll get inside. And some of those are, remember, they're sodiums, they're not 21. Then we have some of those potassiums who are over 21, but sometimes they're like, whoa, it is really getting crowded in here. I think I need to step out for a few minutes. Maybe I'll go out the side exit, or maybe I'll go out in another area. But in the meantime, we've got bouncers, the pump, the sodium potassium pump, right there by the doors. Okay, and this bouncer sodium potassium pump is going to go, whoa, wait a minute, you over there, you're not 21. I know, look at this. This doesn't look anything like you. Don't know who let you in. Let me take, oh, there's 3 of you. I can't believe it. And throw you out. You can't be in here. Meanwhile, it's like, oh, wait a minute. Hey, you two. Oh, you want to go back in? Sure. You are 20. Potassium ions. So we've got 3 sodiums under 21, 2 potassiums over 21 going in. Sodium potassium pump. Okay, now remember that we also have that more negative on the inside, even though we have some chloride ions who are just like, you know, they're hanging on outside and they're not really too concerned or interested in anything. Now, what is that big anion, negativity that stays inside the bar? The bartenders, the maybe waiters, whatever, other people who are washing the glasses and the dishes, and they have to stay in. And let me tell you, they're not happy about it. They're anions, big anions, and there's a lot of them, and they're maintaining the negativity inside the bar or the cell. Okay, I'm going to draw a little, like, you know, just a cartoonish like thing that I'll put up after this. I have a couple things that I'm going to put up as different ways of thinking about this, a little bit, hopefully, that you'll be able to remember, including a little diagram of our bar scene. And then we will be moving on to the conduction of these action potentials. And this is still, the next part's still part of the neurophysiology lecture. I just want to kind of put it in chunks, so it's not quite so much. Okay, so I will be uploading this, and then shortly thereafter, uploading a couple of other ways of visualizing this, and then a video lecture of the conduction of the action potential. Okay, hopefully that helps.